Expertise, mabuhay from the Philippines. For this conference, I am your mentor, Michelle. It's an honor to impart my field of interest with you. This is an overview on the lectures that I will be imparting with you with my next discussion, which is on the sample collection, preparation, and processing for DNA barcoding and geometric morphometric analysis. But before I go through this lecture introduction, allow me to give you a brief background about myself. Currently, I am teaching research and biology at Philippine Science High School Caraga Region Campus, Butuan City, Philippines, and I'm also the Assistant CID Chief for Student Affairs. Going through a brief background of my research journey, my dissertation focused on the checklist and integrative taxonomic study of procuran crabs from selected areas in Mindanao Island, Philippines, combining phonetics, DNA barcodes, and complete mitochondrial genome. This is under the DOST ASTHRDP grant, and I was able to pursue my research fellowship program at Samyang University, South Korea, to conduct the DNA barcoding part of my dissertation. Through the years, my researches focuses on the following field: in geometric morphometrics, taxonomy and DNA barcoding, as well as biodiversity. For this conference, as a biologist by expertise, I am dealing with living organisms and all other biologists can relate on this. And basically, you must understand first biodiversity and conservation. This is very important, not just for the tiny organisms and to the bigger ones or the most intelligent, but all organisms. A collective effort in order to prevent extinction is through appreciation of this field because research is not about the difficulty of the topic but the overall impact it could give on a population. As Neil Armstrong once said, in much of society, research means to investigate something you do not know or understand. The first topic that I will be discussing will be on the sample processing, collection, storage, and information management. In this lesson, you will learn what are your preparations before collecting any samples, as well as after preparations when you have collected, or for storage or for transportation purposes. You will also learn if you will just immediately visit the site and then collect all organism. Is that how you do collection? So for this specific discussion, I'll be focusing on the Philippines setting and you must know your process in your own country. Maybe let's focus on the collection of the true crabs. The discussion will tackle in detail the reconnaissance of the area, as well as the preservation storage of samples, including the process of managing a specimen collection. Permits for the collection and transportation will also be included. These are all important because by abiding the law, you could contribute in preserving any population from extinction. The next lesson will focus on DNA barcoding. So, in this topic, I will be discussing the basic technique on the isolation of DNA from preserved sample and the PCR amplification and nucleotide sequencing, as well as DNA sequence data analysis. In detail, I'll be showing you the importance of DNA sequence on the correct identification of species. As you can see here is an example of Brachyuran or true crab with morphological characteristics similar with the following species, the Atergatis roscius and Atergatis intergerimus. Upon running the C1 DNA sequence of the organism in the NCBI blast, it shows that this is in fact an Atergatis intergerimus. Then the isolated DNA was submitted to the gene bank for data storage with the following accession number. I'll also be teaching the process of submission for the gene bank submission. For the last lesson, I'll be discussing the geometric morphometrics analysis. Let us first learn what is geometric morphometrics. So, as defined by Zeltek et al. 2012, morphometrics is a quantitative way of addressing the shape comparison. It allows us to visualize differences among complex shapes with nearly the same facility as we can visualize differences among circles, kidneys, and letters of the alphabet. In layman's terms, geometric morphometrics is being used by biologists to describe the extent of shape and size variation quantitatively rather than using qualitative method. Basically, this is a mathematical model which gives a shape description, its number counterpart. We can also choose different topic for this specific field. Can either be fluctuating asymmetry or relative warp analysis. I'll be giving you instructions on how to access the software for this specific topic. This is all for the lecture introduction. I hope you will be appreciating the engagement and candor that you all will be contributing to this project. Together, I hope you will be learning from my discussion as well as gain a better understanding about the challenge you will be going through in conducting research. That's all for now, and I hope to see you in my series of discussion. Bye bye.